expertise, including entomology, biodiversity, medicine, public health, epidemiology, environmental science, and more, provides us with such a solid foundation of interdisciplinary collaboration aimed at addressing pressing global health and environmental challenges. So our forthcoming discussion of shared interests and research activities provides a potential for such fruitful collaboration in addressing planetary health challenges. As the name of the seminar states, let's let the research begin. So I'm going to start with greetings from our own University of Illinois Urbana-Champaign Professor Mabokela, the Associate Chancellor and Vice Provost for Global Affairs at UIUC. A few comments, please.
Good afternoon, colleagues. It is always a pleasure to connect with our partners from across the world, and I'm especially delighted that we're having an opportunity to engage and explore together this afternoon. As briefly introduced, my name is Ritu Mabokela. I'm the Associate Chancellor and Vice Provost for Global Affairs and Strategies here at the University of Illinois, and also a professor in the College of Education. And in my current capacity as the Associate Chancellor and Vice Provost, I have the wonderful pleasure and honor of leading our university in matters international, as we often say. And today, I'm especially delighted to welcome you to Let the Research Speak, which is part 2 of our series involving a range of colleagues from our respective institutions in this online engagement. This event has brought together leading experts in infectious diseases, microbiology, and integrative and molecular biology from across our 2 universities. And we really look forward to having the opportunity to hear you showcase your work and to explore how health and disease are impacted by climate change. Today's discussion in particular exemplifies the type of international cooperation that we seek to foster here at the University of Illinois. And when we collaborate with our partners from across borders, we create an environment, we hope, that will enhance the ability of our students, our faculty, our staff, our scholars to have access to richer knowledge, innovation, leadership, and engagement. And as our 2 institutions are both globally oriented, we have a pivotal role to play in shaping how we will respond to our world's most pressing challenges. And it is our mutual responsibility to foster the diversity of voices that should inform those responses. And so as we engage today, I want to encourage and challenge all of us to make the most of this unique gathering of experts and share what we learn collectively from our peers, from our colleagues and others, so that we may positively contribute to our shared vision for a better future. So thank you for taking the time to be here today to engage. And I truly look forward and I'm excited to see what we can accomplish together. Thank you and have a wonderful conversation. Now I'm going to recede in the background and enjoy the conversation.
Okay. Oh, that was Dr. Mabokela. Thank you for being such an inspiration. I thank you so much. And what a perfect transition to hear from the University of the West Indies, Jamaica, Professor Marcia Roy, the Director of the Office of Graduate Studies and Research. Dr. Roy, a few comments from you, please.
Ladies and gentlemen, good afternoon. As Holly said, I'm the Director for Graduate Studies and Research on the Mona campus. I want to welcome our colleagues from the University of Illinois Urbana-Champaign virtually to Jamaica, and thank you for sharing with us in this forum. We hope that one day you'll be able to join us personally in Jamaica. Welcome to all the colleagues and students from the faculties of Science and Technology and from the Faculty of Medicine. Thank you also for joining us to talk about epidemiology and entomology. I want to use my time to especially thank Professor Emerita Celia Christie Samuels, because she is one of our hero female scientists in Jamaica. Professor Christie, thank you for your service to UWI, and thank you for your service to the people of Jamaica and the Caribbean. For your pioneering work in HIV, and in other viruses in Jamaica. When I was putting these comments together, I thought about in March of 2020, at the beginning of the COVID pandemic, that you were one of the first scientists and physicians that was called upon at UWI and by Jamaica to help us treat with this novel infectious agent. And especially, Professor Christy, for your work and your role and your life in helping little Black girls like myself to believe that we can become anything that we want to be, and there are no longer any limitations to what we can aspire to. Professor Christy, what you don't know is that I'm a molecular virologist who have been studying viruses for many years. And it was when I started my research on antiretroviral drug resistance with Professor Figueroa and Russell Pear, and I know Russell is part of your team. It was at that time that the world knew that I was here. So I want to pay tribute to you. I've never met you. We've never had a conversation, but you have impacted little Black girls all over the world with your work, and you don't even know it.
Oh my.
Ladies and
Thank
gentlemen,
you.
she
Thank you
didn't
so
know I was
much.
going to do this, but I thought it was appropriate because
Oh
this is
my.
one of our research heroes in Jamaica.
Thank
And you know,
you.
sometimes we wait until heroes die to recognize them. So I'm doing it this afternoon. Ladies and gentlemen,
Right.
thank you very much. Help me celebrate Professor Christy and the other colleagues who are going to be presenting to you today. Because what COVID taught us is if we all collaborate, we can solve any problem there is. So now I'm just going to shut up and let the research speak for itself. Thank you.
Hardly. Dr. Roy, I kind of heard you invite us all to Jamaica, so I'm going to hold you to that.
All right.
What a joy that would be to meet all of us, eventually meet in person, but to visit your beautiful country. Dr. Roy, you said something so interesting. You talked about heroes, and I think I have the very great privilege of introducing heroes going forward. So going ahead, I'm just going to give a brief bio for each of you, barely touching the surface, just highlighting, and only more limited by time, but highlighting people's impressive research and work. Just from a housekeeping standpoint to our wonderful audience, put questions in the chat. We'll try to get to them at the end and then follow up. And it just as a friendly reminder to our heroes, we will limit remarks for about 10 minutes. So I'll be keeping an eye on the clock. So our first speaker we'll start with is here in Central Illinois, Dr. Brenda Wilson. So Dr. Brenda Wilson is a professor of microbiology in the School of Molecular and Cellular Biology and the Carroll Illinois College of Medicine. And her current research work focuses on host microbe interface, bacterial pathogenesis, bacterial protein toxins, development of toxin-based therapeutics, biogenics, genomic technologies, and applications involving microbiomes and their roles in health and disease. She's also looking at climate change impacts on health and microbiomes and the development of technologies and applications for detection and risk assessment, among many other things. Dr. Wilson, please proceed with your comments.
Thank you so much for the short but very nice introduction. I appreciate that. And well, it's a very big pleasure for me to be here today, and I really appreciate all of the enthusiasm for building some, connections between our two institutions, and also globally. Just the idea that we are expanding our connections is really exciting. I'm going to be wearing kind of two hats here, both as a researcher as well as an educator, and talking about some of the activities that some of us have been, even that are on this call right now. So Dr. Holly and Dr. Jaffia, as well as Trent Ford, who spoke at the last meeting. We've been actually working on some collaborative projects. And so I'm going to share my screen right now and talk a little bit about some of those activities that we've been doing. And I also want to emphasize that putting on my educator's hat here, we have a lot of very shared strategic priorities that we would like to also build stronger and bolster through some of these activities that we have with y'all. And so we would like to excel. Of course, we want to excel in research and creative activities. That's the whole point of the research, and we are very excited, and that's our backgrounds, and that's what we would like to promote. But we also want to, during that process, enhance educational experience for both our students and our faculty. And we feel that we can do this by strengthening our resources and the visibility and impacts that we have at all levels of our education, academia, research, and just community involvement and engagement. And of course, we're very, very interested in promoting diversity, equity, inclusion across, again, all levels. And we're doing this in the group that I mentioned. We're a little bit bigger than just a few that I mentioned, but what we're trying to do is actually build a network for integrated climate, health, and environmental systems. And we're calling this niches. And basically what we feel is that food has a very big impact on us, and it does so through multiple ways. So it impacts the environment, and that of course impacts the nature and the quality of the food, and that interacts of course with our microbiomes. And we're all microbiologists. I'm a microbiologist, and so I'm very much focused on looking at things like the microbiome and its connectivity to health. But all of these, I think, are very closely related to each other. And what we are trying to do is invite community educators to join us in advancing scientific literacy and enthusiasm for science by engaging in research and learning partnerships that involve One Health, microbiomes, and regenerative food systems that address things like food insecurity as ways to build resilience. So we all know that climate is impacting us, and it's very difficult to change climate. But what we can do is build more resilience to those changes, and we think that we can do this by bolstering our environmental and human health through mechanisms that will make us healthier. And so our multidisciplinary team, we call it the Niches team, is made up of folks at the University of Illinois, both in the College of Liberal Arts and Sciences, the School of Molecular and Cellular Biology, of which I'm Associate Director of Education at, and as well as our very new but exciting Carle, Illinois College of Medicine, of which Holly and Jaffe and I are all members. And then also the Illinois Extension, which is a very expansive outreach program that we have in Illinois to reach communities and farmers and educators and the whole entire community about the things that we do. And then in terms of our focus and what we're trying to do here is we believe that education empowers all stakeholders to make informed decisions about their health and well-being. And we are proposing, in part of the work and the approach that we're trying to take, is that microbiomes can serve as indicators for overall health of both soils and the environment, if you will, as well as human communities. And that when we have healthy agricultural and food systems that are supporting healthy communities and resilience in response to adverse climate events. And these healthy microbiomes will prevent some of the diseases. They will protect against the impacts from climate change in this fashion. And so what we are looking for is building partners, academic partners to help us advance educational and training modules, looking for curricular development that embraces the ideas of One Health, food security, nutrition, and health impacts that will allow us to actually be able to handle things that climate change imposes on us. And then we're looking for our community and global partners that will also help with experiential experiences that are immersive and that really make high impacts on those that are engaged in this activity, both from the students as well as the teachers' point of view. And we are hoping that in all of these, we're going to be generating future citizen scientists that will help with overall community scientific literacy. And of course, we really are engaging our clinical partners for being able to build, future, workforce that actually helps with understanding how to promote health in ways that we are being more preventive rather than reactionary. And of course, this involves training and continuing education for all levels of education, going from pre-K to undergraduate to graduate to medical students, other educators, training the trainers sort of thing, and practicing clinicians and continuing education. And so at Illinois, we have a lot of various research opportunities available. We have a very expansive program in this, and especially in the School of Molecular and Cellular Biology, which I work with the undergraduate education. We have 2,000 majors within our school, and we, of course, deliver courses for on the order of 12,000 students every year. We feel that we are a leader in research, in teaching, and in public engagement, and we really strongly feel that this empowering new leadership happens by actually developing research, education, and immersive experiential learning opportunities. And we very much value our education and partnering with not just local, but also global communities in furthering science education. So our initiatives team is aiming to invite folks to join us as community and global partners to build strong, vibrant community collaborative networks that are interdisciplinary in all aspects of this: research, education, and workforce development. And with these particular goals in mind, that will help strengthen food security, nutrition, and thereby mitigate a lot of the health threats due from climate change. And this is just a smattering of some of the work that has been done with some of the folks in our group. And our objectives is really to look at, in terms of the research that we're trying to do, is look for causal linkages between climate change and human disease. One of the major challenges that we have had in the area of looking at climate change and health is that the data there's a lot of data, but we don't have really good correlations that pinpoint and actually say, yes, this is the cause of whatever that health impact is. We see effects, but we haven't actually got really good, strong data for that. And what we're trying to do is remediate that. So our objectives is to collect, assimilate, analyze, reanalyze, and interpret large sets of data that is already existing in terms of climate, in terms of epidemiology, as well as clinical health data, integrating all of those to generate a valuable data set and insights that will actually support some of the models that we have regarding climate change and its impacts on health. And then we would like to, of course, develop predictive models based on all of that, and then test them. And so that's part of our research. And this is just a small example that was done by a student in bioengineering under the guidance of some of our faculty preceptors that are in the College of Medicine. And what they were doing here is looking at mapping causal linkages between practices, agricultural practices that pertain to regenerative agriculture, and then colorectal cancer rates. So the instances of colorectal cancer in particular regions. And this was focused mostly in the Midwest. And what they basically are doing here is looking at a regenerative agricultural index that was defined based on 13 regenerative agricultural practices by county. Then they were correlating it with the health statistics regarding colorectal cancer rates in each of the counties. We know there was a gap here, but several of the ones that are in these states were participating in this activity. And then when you overlay these two factors and look at what the index is for them based on correlation of the RA index versus the colorectal cancer rate, when you have a high rate of regenerative agricultural practices, you actually have a reduced incidences of colorectal cancer. And so this is a correlative, and of course it allows for one to start building a model that can then be further tested by looking at more specifics and looking at other data and setting up systems for testing what aspects of these regenerative practices that contribute to the reduced colorectal cancer rates. We've also we're looking at, and this work, we had some help with Trent Ford on doing, where we looked at the some climate models for the both past and generating what would be trajectories for the future, and we were then correlating that with what we have in the United States called a social vulnerability index, which has at least 16 or so different factors that contribute to it. And basically it points out, and where you have the darker, colors, what you end up with the highest risk for social vulnerability, and economic vulnerability. And here we just look at focusing on Illinois, and in particular Champaign, near where we are. You can see that we're not exempt from having some areas that are particularly troublesome. And then what we're looking for is correlating that with climate, making those models, if you will, looking at the particular communities that we have in these models, and then deciding if we provide an intervention, and those interventions can be in the form of many different aspects. But if you do it on a more global scale of education and training and practices and things that you use on a large community impacts, that if there is an intervention strategy that you introduce, the idea is that if you look at two different communities, one that might be at low risk versus one that might be at high risk, and then you introduce that intervention, you would expect that you'd be able to start discerning over a period of time, whether or not those interventions are being effective or not. And so possible outcomes from this research would be making more informed decisions at the government level, at the educational level, at all kinds of different aspects in health and insurance, et cetera, in terms of being able to make those policy changes that are needed. And then also looking for what types of educational and training interventions need to be implemented in these cases. Another aspect that we're looking for is we, of course, come from a more microbial and microbiome perspective, and we are collaborating with the University of Chicago, Illinois. One of the professors there is a member of the Discovery Partners Institute, and this is an innovation hub within the University of Illinois system that has, and Dr. Rachel Peresky has a vast archive of samples that were collected beginning around near the end of the COVID period for looking for at sewer and wastewater treatment plants, and finding in those wastewaters COVID. So it was started out of a lot of the research that was initiated during the COVID, and collections and infrastructure that was put into place in collaboration with the Illinois Department of Public Health and the Discovery Partners Institute in the University of Illinois, and all basically setting up this wastewater surveillance system. And there are many collection points all throughout Illinois, including 2 that are located here in Champaign-Urbana. And this is just an example of some of the output that has come from those efforts focused from the 2 centers that were in Champaign-Urbana, looking at both collections that are in response to COVID, and now the more recently the concerns with RSV and flu, and so.
Dr. Wilson, I'm
Yes.
so sorry. This is so exciting. This research, these correlations are fantastic. We're just a bit over. I would
Oh
maybe
yeah. Okay. I'm sorry.
Yeah, I'm so sorry. I need to
No
move on.
worries.
Your research is so exciting. Thank you. If you could just
I'll wrap
tie up
up right
a little
now.
bit and
Yeah,
then
because this is part of
thank you. I
this
just
is
want to move
part of
on
what
to
we want
our
to
next
do.
speaker. Okay,
Right.
sorry. We can listen to you for hours. I'm seriously.
Okay. All right. Well, what we're trying to do is reach out to collaborations that might help with very similar types of research and looking at using microbiomes and even expanding this not to just viruses, but also to other types of pathogens, and being able to do these kinds of analyses longitudinally to help build some of the models and support or confirm or adjust some of the models that we actually get out of this. And we'll be very, very pleased and happy to have some folks to collaborate with and do some of these kinds of studies for us. And I'll end there. Sorry for going over.
Oh no, thank
It's
you,
easy.
Dr. Wilson.
It's
We can
easy to
listen
talk
to you all
about these
day.
things. Yeah.
You
All
know,
right, thank
I
you.
think the theme of partnership really came through strong. Such wonderful partnerships, the potential for such wonderful partnerships. Thank you so much. I'm going to move on to our next hero. I'm stealing this from our Dr. Roy. Our next hero is Dr. Cecilia Christie, who we heard a lovely, lovely introduction. As Dr. Roy shared, she is a professor of pediatrics, infectious disease, epidemiology, and public health at the University of the West Indies, and with a robust background in infectious disease, public health from University of the West Indies, as well as Yale and Hopkins. Lots of exciting research with clinical trials on vaccines, emerging infectious diseases, and leading to numerous publications and awards nationally and internationally. Dr. Christie, please share your comments.
Share. Oops. Insert. And to share. Oh. Good afternoon, everyone. It is indeed my pleasure, my privilege, and my honor to join in this collaborative roundtable discussion between UWI and UIUC. And thank you so very much for your very warm and kind introductions. My topic this afternoon will be about dengue, Zika, and chikungunya, the clinical epidemiology of these 3 viruses, these 3 arboviruses in Jamaican children, as this is linked to climate change and climate health. So I'm going to essentially speak to dengue in the context of comparing the 2018-2019 epidemic, which was a serotype 3 epidemic, to the 2023 epidemic that we are now undergoing. I'm going to have a few words on chikungunya and Zika, and then I'll speak to and make a few comments about new drugs and vaccines. Here we have the Zika Action International team at our kickoff meeting here in Jamaica in October 2016. And essentially, we did quite a bit of work with these 3 viruses under funding by the European Union. We won't be talking much about that today, though. We'll just be talking about what I just said. So dengue, we know, is the most important arbovirus, and we have 4 serotypes. And 1 serotype increases disease severity with reinfection of subsequent serotypes. And children, we know, have the most severe presentations and certainly the highest attributable mortality. In 2019, dengue was listed in among the top 10 threats to global health. And in 2023, the numbers reported cases surpassed the 2019 reports by over 1 million. And there was significant attributable morbidity and mortality. Here in Jamaica, we have been tracking dengue epidemiology now for almost 50 years. And certainly we have seen that the dengue epidemics are occurring in more frequently and with more intensity. And in 2014, the epidemic was overridden with a chikungunya outbreak. And in 2016, not only did we have dengue, but we had chikungunya and we also had Zika. When we look at the seroprevalence in our pregnant women, you see that most of our women, if not all, are seropositive for dengue. About 84% are seropositive for chikungunya. But for Zika, that's 16%. It therefore means that Zika can be introduced in our country, reintroduced at any time in the future. Looking at the dengue trends in Jamaican children, comparing the two periods of 2018, 2019 to 2023, the serotype 3 versus serotype 2 epidemic, you'll see again that children less than 15 years of age accounted for a great deal, a great proportion of the cases that were reported nationally. And of the 86 deaths in the 2019 epidemic, 62% of them were children. And as of now, last month, 45% of the deaths were children. The clinical syndromes that we recognize in children in particular include a severe undifferentiated febrile illness or non-severe dengue. And then we pay attention to dengue with no warning signs, which in general is sort of managed in the outpatient setting. And then dengue with warning signs, we admit these children to hospital. And of course, those with severe dengue, which is characterized by plasma leakage, fluid retention, severe bleeding, and severe multi-organ impairment, which may involve the heart, the lungs, and the brain, and also the liver. We admit these children. Several of them, of course, will be managed for shock and also for bleeding. And if these children are not managed appropriately, they will die without intervention. So looking at our data from the 2018-2019 epidemic of some 340 children that were admitted to 5 of our area hospitals, you'll see where 60% or more had warning signs and 20% of them had severe dengue, which was hemorrhagic for the most of them. Hepatitis, hemoglobin SC disease was associated with severe hemorrhage, dengue with hemorrhage in this epidemic. Organ system involvement occurred in over 94% of cases. And for those who are admitted to hospital, over 5 to 7 organ systems were involved for a significant number of persons. The gastrointestinal system with hepatomegaly, the hematological system and the clotting system in over 90% of cases, cardiac involvement in 26%. And we also had children with respiratory, kidney, and central nervous system involvement. The hematological involvement was rather significant, and essentially over 80% of them had severe thrombocytopenia and 50% of them severe leukopenia. They also had a significant number of very, very high hepatic enzymes, and sometimes it exceeded over 120,000 international units per liter, something that I've never seen before. And where these numbers were very elevated, these children tended to have severe dengue. For those who died, over 94% of them had severe dengue, 88% of them had hemorrhagic dengue. There was a significant number who had comorbidities, and hematological and gastrointestinal involvement was uniformly involved in all the children. And although 26% of the total cohort had cardiac involvement, 77% of the deaths were cardiac. There was also shock and hypotension and severe involvement in the blood system and severe derangement in various bleeding derangement as well too. For those who presented late, essentially regardless of whatever we did to resuscitate them, the mortality was significant in the first 24 to 48 hours. And then along came the dengue serotype 2 epidemic, which we are now trying to document. The dengue serotype 2 epidemic, we know, was associated with antibody-dependent immune enhancement, with more severe outcomes in children in Nicaragua, where over 3% of those that presented actually died. This followed recent exposure to Zika and at least one prior dengue infection. So we had our antennae up because when dengue serotype 2 was circulating here, we wondered if this would be very severe, if there would be more severe manifestations, and especially bearing in mind that there were quite a few current circulating viruses as well. So we have not seen dengue serotype 2 in Jamaica for the last 14 years, and the last epidemic was way back in 1968. And certainly it has not circulated since Zika circulated in 2016. So what have we seen here at the University Hospital? During the September to December, we were seeing in the outpatient department over 800 children being seen there per month. The majority of them were being managed for dengue without warning signs, and they were being managed at home, even though a subset might have had leukopenia, because we were worried that we wouldn't admit them because they might have secondary infections because their white cell count was down. For those who were hospitalized, we had over 80 admissions. I think I took care of about 40 of those in October when the epidemic was at its highest. And these children presented mostly with dengue with warning signs or without warning signs in those who had underlying diseases. What was interesting is that although we were expecting severe dengue, there were none who presented, none of the 80 had severe dengue, none had hemorrhagic, severe hemorrhagic manifestations, or none presented with shock, which is rather unlike the 2018 epidemic. Despite that, though, we saw quite a few unusual cases. So, for example, we saw neuromyelitis optica spectrum disorder, which is a disorder where children present with paralysis and blindness. And I couldn't find any cases like that in the literature. I found 2 related to dengue in adults, but none in children. We also had a few deaths. There was a child with influenza co-infection with sickle cell disease, and this child had multisystem involvement with the hemophagocytic lymphohistiocytosis, where the essentially what happens is that the red blood cells eat up the white blood cells, and you have that multisystemic presentation. There's also a child who presented with fulminant myocarditis with high cardiac troponin enzymes, which were way over 7,000, and had ventricular tachycardia, which was unresponsive. And this child died in the ICU within 36 hours of initial presentation. Dengue has been occurring recently along with other arboviruses that are also related to climate change. We remember the 2014 chikungunya outbreak. Here in Jamaica, it decimated the whole island. We had widespread absenteeism from school and work, loss of productivity, economic losses that were huge, and a huge health sector impact, which also affected the children. And there was increased need for bed space, pharmaceuticals, diagnostics. When we looked at over 200 children in 2 of our hospitals who were hospitalized, 93% of them came from homes where there were several family members who had chikungunya. The majority presented with fever, maculopapular rash, and joint pains. The little babies presented with irritability and loud groaning. Those who were 6 months to 6 years had febrile seizures, and a significant number of them had the central nervous system involved. Here you see some of the little babies with joint manifestations. You can see joint inflammation. You can see skin involvement with maculopapular rashes and vesicular rashes in various stages of development. And then along came in 2016 the Zika outbreak, which not only affected us in Brazil, but right here in the Caribbean and in fact right through Latin America. And here in Jamaica, we were not spared because, again, we had quite a few children who presented with microcephaly. And here we have one child here, and the microcephaly rates were approaching up to 3% in some of our hospitals, in our Kingston and St. Andrew hospitals, where we were following these babies. And essentially, you'll see here where the features radiologically and clinically fit in with the Zika syndrome, congenital Zika syndrome. And then the children also presented with rashes and fever and headache. And the rashes, you'll notice, did not spare the palms or soles. It was maculopapular, and it also involved babies. For those who couldn't complain, the majority of them had headache, they had red eyes, they had joint inflammation, and so on. We also had quite a few children and adolescents who came in with paralysis and evidence of neuroinflammation involving the spinal cord and also involving the brain. And quite a few of these children also demonstrated household exposures to family members who had Zika. Of course, we know that we do not have any confirmed treatments for any of these viruses that are appropriate. But here on the island in Jamaica, there are some things that we could use if we were clever enough to implement them in clinical trials. So we know that papaw leaf or the carica papoya leaf has been shown in 3 randomized controlled trials to improve the platelet count in patients who have Zika, and it's also a pro-inflammatory cytokine. Vitamin E also has been shown to show faster platelet recovery and reduced liver enzyme in derangement. Ivermectin, which is that controversial antiviral agent, has been known also to show in patients with dengue faster NS1 antigenemia clearance. And vitamin D, which is also been shown to reduce viral replication and inflammatory cytokine production in vitro and reduce the relative risk of dengue hemorrhagic fever. There are certain vaccines that are available, but none of them are approved for use, certainly not here in Jamaica, so they're not available to us. And I just need to make a plug that while we are ongoing a dengue serotype 2 epidemic, we're also doing this at the time of SARS-CoV-2, COVID-19, which is also circulating here. The Omicron subvariant has been circulating for quite some time. And also we have a pretty significant influenza and RSV outbreak simultaneously. So the question would be, quite a few of these viruses may present in the same way, or could they present in different presentations, or what is the role of virus co-infections and interactions?
So Dr. Christie, I'm so
finishing.
sorry, we can listen to you all day. So just give you a little
this is my last slide.
Thank you.
And this is my last slide with the ladies from the Caribbean who presented at the Zika 3rd International Conference in Washington on varying aspects of these viruses. Thank you very much for your attention.
Dr. Christie, what a stunning overview of these clinical presentations, especially in the devastation in these children. I also think it's fascinating some of the therapeutics and the novel therapeutics. I'm so grateful for this. This is absolutely wonderful. Thank you so much. We could listen to you all day as well. My gosh.
Thank you, ma'am. Thank you.
Thank you so much. It's so hard to move on. I'm watching the clock and keep stress. So we're going to move on again. And I just again, thank you for such a stunning and overview of that. So I'm going to move on to my colleague, Dr. Jafia Ramkumar, a board-certified internist and a clinical associate professor at Carroll Illinois College of Medicine. She also at the American University of Antigua, and she's an experienced medical educator. She focuses on the triple crisis of climate change, biodiversity loss, and pollution, and helps develop medical school curriculum, including curriculum for the community, locally, nationally, and internationally, on topics of climate change, health, and health impacts through a One Health lens with a focus on nature-based solutions. More recently, her work has included the farm, food, and health connection through microbiomes and regenerative agricultural practices. Dr. Ramkumar.
Thank you, Dr. Rosenkrantz. So, good afternoon. First of all, I'd like to say that as a proud graduate of the University of the West Indies, undergrad and med school, I am delighted to be here to be participating in this joint session and share the work that I've been engaged in at UIUC. I'd also like to add that much of this work around climate change, planetary health, and sustainability is done in partnership with my colleagues, such as Dr. Rosenkrantz, who's our moderator, Warren Levy, and Dave Ramkumar. You may have seen their names scattered through previous presentations. I'm going to outline this journey of just riffing on the title of this session as it relates to working in spaces of education, community engagement, and research. So much of our work is really focused on medical education as medical educators. And in a world where we're facing multiple planetary crises, such as climate change, biodiversity loss, and pollution, healthcare providers and other learners really need to be prepared to deal with these global public health threats. And we have done this by trying to educate learners about the impacts of these threats on human health. And very importantly, trying to make the connection of the whole interconnectedness of the health of the planet to the health of ourselves, of humans. And we do this with a One Health approach, and really positing that by protecting the health of our planet, we are ensuring our own human health and well-being. And I'd like to add that we really try to emphasize an environmental justice lens in all of our work that we do. So I am a master gardener, and I've had this lifelong passion for environmental conservation. And as I was thinking about solutions to these big, wicked problems, one of my areas of focus is nature-based solutions. And as I see it, nature-based solutions are health solutions. I mean, we can literally plant healthy air and clean water. We can tackle the epidemic of obesity and chronic non-communicable diseases by promoting, for example, a plant-forward whole food diet, which also happens to be more sustainable. And what if we grow and produce our food with climate-smart regenerative agricultural practices, as Dr. Wilson had elucidated in her presentation, instead of our current unsustainable destructive practices? It would be a total win-win for people and the planet. And so our journey began several years ago with this single lecture where we're looking at the health impacts of climate change. And as we were looking for more ways to incorporate climate change into an already packed medical curriculum, and everybody knows how hard that is, trying to get things into a packed curriculum, we decided that the best way would be to try to add content into existing content and we did this through simulation. And so we created an objective structured clinical examination, which the third year students in their internal medicine clerkship, this is what they did. So we didn't take any extra time to do this. And we created a standardized patient scenario where there was a patient whose asthma symptoms were getting worse because of exposure to smoke from wildfires. And unfortunately, this is becoming more common. And in the debrief with the students, the facilitator would then have the opportunity to discuss the environmental drivers and would open up discussions for steps to mitigate for this, to have strategies for mitigation for this patient in ways that would go beyond the normal treatment, or as we would like to say, beyond the inhaler. And we studied the impact of using this OSCE, as it's called, to integrate climate change into the curriculum. And we got very positive results. And this spurred us to publish the case in MedEd Portal, which is a journal for teaching and learning resources of the Association of American Medical Colleges. These are some other workshops where we've deployed different pedagogy. And they've included having students actually create their own mini scenarios using patient and community case studies where they would discuss climate change threats and impacts, adaptation and mitigation strategies. We would have patient care workshops in which the learners develop their own patient climate-related cases. And during the COVID-19 pandemic, we used that opportunity to really examine the syndemic of COVID-19 and the climate change crises. And as always, we really tried to do this with an environmental justice lens. So, we particularly want to focus on the poor, the minority, disabled, and elderly people who are affected primarily and predominantly by climate change and COVID-19 when it happened. So later in our journey, we decided to pivot from patient care to advocacy. Because we didn't just want to raise awareness and increase knowledge about climate change and health. We wanted our students and our learners to be able to do more than that, be able to use their voices to advocate for meaningful change. And so we developed this workshop in advocacy, and it was sort of like a primer in advocacy. And in these advocacy workshops, our learners developed legislative presentations on climate-related bills. So for instance, it would be a bill related to children riding diesel-powered school buses or urban heat islands. So these were just examples of things that we did. Again, we studied the impact of using these kind of advocacy workshops, and we published it in MedEd portal. The next step in our journey was development of a 2-week elective on climate change, planetary health, and sustainability. And here we really focus on the multiple impacts of climate change on human health, as you can see with the CDC wheel. And what we also did was we explored different aspects of nature-based solutions and other strategies for adaptation and mitigation. And because it's an elective with few students, we really worked to customize the course to the interest of the students. And because Illinois College of Medicine is an engineering-based medical school, we really worked to incorporate innovativeness throughout the course and through assignments. Next, we broadened the reach with an interprofessional One Health session, which brought together medical students, veterinary med students, and public health students. And we brought them together to discuss the impact of diarrhea from a contaminated water source from a flooded cow manure lagoon due to an extreme weather event. So this was a scenario that we set up. And it was a very successful way to integrate climate change, human and ecosystem health impacts into the curriculum. Again, we studied and published this as well. Now, our collaborative efforts have continued, and we've actually developed a transdisciplinary team that really works to shift paradigms to a more regenerative and sustainable food systems. And this project that you're looking at right here, the Regenerative Agriculture and Human Health Nexus in the Age of Climate Change, its goal was to promote the concepts of farm, food, and health and that whole interconnectedness. And these connections are essentially about healthy soils producing healthy foods for healthy people with regenerative agricultural practices. So that's really this project in a nutshell. And this slide that you're looking at here summarizes this whole concept of the connection of healthy soils for healthy plants and healthy people. And the through thread is really the microbiome. So you can see on the left in red that when we're with conventional agriculture, where we're destroying the soil ecosystems and microbiomes, we're producing unhealthier plants, and if we're eating it with an unhealthy diet, then we're making ourselves and our gut microbiome more unhealthy. And the flip side on the right side is with healthier soils, producing healthier foods that we're eating with healthier diets that produced through regenerative agriculture, then we are promoting better health for us. And this work on the farm food and health connection through the microbiome has produced many lectures, presentations, and workshops. And actually last year, we developed this very novel, innovative elective, which looked at the food systems in a holistic way. And we had faculty from multiple disciplines across the university provide their expertise in areas that students would never normally be exposed to. So things like animal ag, regenerative ag, food systems, nutrition that would not be normally taught in a medical course. Microbiomes, economics, policy, and culinary medicine. The students were exposed to all of these things in this elective. And as I was saying, culinary medicine is a huge component of these workshops and the elective that we did. And they really center around cooking gut microbiome friendly meals. And we've deployed these sessions in several venues, from Illinois College of Medicine to the University of Illinois Peoria Family Medicine Program, to the American University of Antigua's Medical School. And this elective the microbiomes matter, path to regenerative systems of farm, food, and health in the age of climate change was so successful, and the students were so captivated that they wanted to write up about their experience and share what they learned. And they did, and we actually just got published in the journal Challenges earlier this year. This slide just is a very small example of some of the lectures that we have engaged with across the university. So we've engaged in looking at climate change through a biomedical ethics lens and through a biosecurity lens. And now I'm going to just pivot finally to show you some examples of community engagement. Here we've done multiple community talks. I've just used an example here where I did a talk for Earth Day in Kingston, Jamaica for Environmental Solutions Limited. Dr. Rosenkrantz did a talk on climate change in your backyard for the Rotary Club in Urbana. With this slide, what we here, we have engaged, this is our engagement in the Unit 4 school districts. And here we've engaged with students and teachers and administrators. And I think Dr. Wilson was really talking about this with some of those niches slides that she presented earlier. And so it's all about that connection to planetary health, human health, the farm, food, and health connection. And it's really about pushing back against and creating a counter narrative to the epidemic of processed and ultra processed food that we know our students are inundated and surrounded by. We've also worked in community gardens, creating biodiverse habitats and growing fresh produce. And some of the, you'll note in the middle, this climate change in your neighborhood. This is a UIUC student research initiative, and some of our faculty here that are here on this talk and in the previous session are faculty advisors to this project. And what this project is looking at is the differences between spaces with native vegetation and mowed spaces, and by looking at its impact with climate change, by looking at metrics such as air quality, temperature, soil quality, in particular looking at microbiome analysis. So the point of this is to perhaps hopefully get the data that will support more urban green spaces. This slide is about an interpretive sign that Dr. Rosenkrantz and I collaborated with the Champaign Parks District, Urbana and Champaign Parks District through the Champaign County Medical Society. And really, it was about promoting the health benefits of being outdoors in nature. And this was it being installed in one of the two parks, one in Champaign and one in Urbana. And I'm going to wrap up pretty soon here. This is a slide that's looking at some occasions where we were involved in advocating for various policies and legislative bills that would mitigate the impacts of climate change. So in the middle, this is Dr. Rosenkrantz and Warren Levy meeting with our state senator, Tammy Duckworth, trying to promote climate-friendly policies. And we've talked about, we met with local and other state legislators to promote things like the Clean Energy Jobs Act and climate-friendly ag practices. So, and this is also, we work closely with Illinois Clinicians for Climate Action and Champaign County Climate Coalition, which are organizations that Dr. Rosenkrantz and I are very actively involved in. And lastly, I just want to end with to share some of the ongoing research opportunities. And they're the ones that you're seeing here in red that we're currently working on. And many of them we are working in collaboration with some of the speakers that you met on the previous session and some who you're meeting today. And in particular, we are very excited about this idea of connecting health outcomes to climate data and climate change and urban green spaces. That was certainly that was a theme that was brought up in the last session. These other two topics, agricultural practices, food, and the effect on the gut microbiome, particularly relate to this food, farm, and health connection.
This is great. Oh,
so I'm going to wrap up. I'm done.
Okay, so we'll have,
So feel free to
perfect.
contact me.
I keep grabbing you guys on your last slides. Okay, thank you very much. It's been a real journey. I have to say I've shared so much of this journey, and it's been a joy, and it's just really evolved into places that we could have never imagined. So, thank you for sharing the journey and so much of your thoughtful innovation. Moving forward, moving beyond a single lecture and one where we provided lunch. We've come a long way. I'm going to move on now. We'll talk. Our next speaker is Dr. Milton Tan. And Dr. Tan is an assistant research scientist at the Illinois Natural History Survey and the Prairie Research Institute at the University of Illinois at Urbana-Champaign. And his research focuses on the integration of genomic and evolutionary analysis for understanding the origins of biodiversity, including reconstructing the tree of life, understanding drivers of evolutionary diversification, and the genetic basis of phenotypes. He's interested in expanding his collaborations to apply genomic and evolutionary techniques to diverse organisms. Dr. Tan.
Thank you very much. Thanks for that introduction. And yeah, thanks for giving the opportunity to speak today and meet a bunch of new folks. Yeah, so today we're gonna talk a little bit about my research in biodiversity genomics at the Illinois Natural History Survey. Research at the Illinois Natural History Survey has focused historically a lot on understanding the biodiversity that's out there. What are the species that are out there? How are they doing? And documenting biodiversity, of course, is very important for understanding what is out there and what is to protect and how that might respond to future scenarios such as climate change. And it's clear from ecological and evolutionary research that there's lots of species, lots of populations, and that their diversity is shaped by their evolutionary history. And of course, all life shares the usage of DNA. And so I apply genomic techniques to better understand what species are out there and how they've evolved, what are their traits, and better inform how we might be able to manage and conserve species. So while my title is ostensibly a biodiversity genomics scientist, I particularly focus on fishes, which represent half of all vertebrate species. So I've studied everything from some of the smallest to the largest living fishes, from minnows to sharks. And because genomes harbor a record of evolutionary history and encode the toolkit that organisms use to survive, genomics can answer a variety of questions. And understanding what species are out there and how they've evolved and diversified could help understand how they might also respond to future threats. And so I'm going to talk about some of the examples of types of questions that we approach in the lab. And again, these are just examples in fishes, and I think that there are opportunities to apply these questions outside. So for the vast majority of species, we still are trying to understand even what they are and where they are. We can better understand what with genomics, sometimes species that are harder to identify. One of the questions that the Illinois DNR had for us was what species of cavefish that we have. And this is a pretty difficult question because these are fish that live most of their time underground and only come out of from underground a few months out of the year. And these are a threatened species in Illinois, but some recent work had suggested that there might be 2 species of this cavefish. And we didn't know which one we had or whether we might have had both. So using genomic techniques, we were able to sample a variety of these cavefishes, and we determined that these cavefishes in Illinois represent a single species, the Shawnee Hills cavefish, which means that we only have one species of conservation concern here to protect as opposed to 2. And so genomics is very useful for these kinds of identification of species for which we don't have the specimens in hand for us to look all over to identify them. But populations are not also identical across their range. Populations can be structured and different populations can be separated from each other due to barriers. And this can be important to understand because when we're trying to manage populations, some populations are not genetically interchangeable. There might be local adaptation, and moving individuals between locations might not work as well if certain populations are locally adapted. What we found using genomics was that the Illinois populations of this particular cavefish appear to be genetically distinct from their closely related populations in Kentucky. And so again, genomics is very important for helping us understand what we need to do to help manage and protect this species of conservation concern. Another really interesting aspect of genomics is that it provides us the potential to even estimate population sizes. And of course, population sizes are a very important aspect to consider when conserving organisms as well, and understanding how they have previously responded to climate scenarios or other stressors, as well as understand the genetic diversity that they might have still available to them to adapt to future changes and challenges to their populations. And so using these kinds of genomic tools, we were able to estimate that population sizes in this cavefish apparently were larger in the past, but that there were declines, but that these declines happened maybe about 1,000 years ago, probably prior to when human impacts to groundwater, for example, might have been occurring. And so there seems to be a more recent stable trend in their population sizes given these genomic data, which again are a really hard to survey aspect of these fishes that primarily live underground. And so genomics can be a very powerful technique for us to understand how species are doing so that we can better manage and protect them. Now, most of my research actually focuses on reconstructing the tree of life and understanding how that provides predictions for how traits have evolved, how species have diversified, why certain groups of species are more diverse than others, and how they've evolved and adapted to challenges over evolutionary history and geological timescales. And here we're looking at an evolutionary tree of one of the most diverse groups of fishes, the carps and minnows, which number over 4,000 species. And using this evolutionary tree, we were able to estimate these variable rates of diversification across the tree, with red indicating more rapid rates of evolution or speciation, and potentially decreased rates of extinction. And these could potentially later be correlated to aspects of environment or their traits that might allow us to better understand what helps to these species buffer against extinction over at least evolutionary timescales and project forward into the future. Similar questions asking about the influence of the environment on diversification and evolution. In this PNAS paper that I was involved in that was published last year, we were interested in understanding the diversification of South American fishes. And by reconstructing the evolutionary history and understanding the relationship between the movements of these fishes across South America, we were better able to understand how past connections of rivers helped to increase the diversity of fishes in the Amazon, resulting in this being one of the most diverse places in the world. I also used these evolutionary techniques to understand how species evolve. I was a part of sequencing the whale shark genome, and the whale shark is famously the largest fish species. And it's not just big, but we're using these evolutionary analyses, we found that it evolved its body size roughly 4 or 5 times faster than other cartilaginous fishes. So it's surprisingly large compared to its close relatives. And this suggests that there might actually be kind of correspondingly unexpected genomic changes that have evolved in correlation to this gigantism, if you will. We did a lot of analyses, but I'll focus on this one in particular. We compared the whale shark genome to that of other small and large-bodied vertebrates, such as elephants and baleen whales. Now, large organisms, paradoxically, they have more cells and therefore more cell divisions, but don't seem to experience higher rates of cancer, which is something that you might expect if you have more cells. And so previous research have suggested that that large-bodied vertebrates must adapt in their cancer suppression genes to avoid cancer. And this appears to be the case in the whale shark as well, although different genes in the whale shark compared to other species. And so this seems to be a common pattern that we were able to study using this kind of genomic tools. I'm not going to talk about a particular study here, but genomic tools, genomes are not set in stone. Genomes also respond to the environment, and I'm very interested in expanding research using changes in gene expression, seeing how the genome functions to understand how, for example, individuals or populations or species might be responding to environmental changes such as diseases or climate change. And this can help us better understand how to manage these species as well. So I think that there are some potentials for collaboration with this type of research. While I talked about fishes, there's a lot of different types of species that these techniques could be used for. I've been talking with Chris Stone, who talked last time, about applying these techniques to better understand how mosquitoes, which are vectors for diseases, are moving about and how they are expanding their populations across the country. And so these techniques might be useful for better understanding these mosquitoes and any species. people that are studying environmental stressors and how organisms might respond to them in their fitness or their physiology or their ecology might also be able to incorporate genomic tools to understand how gene expression changes with respect to these different kinds of scenarios, and potentially better understand how species are able or not to adapt to these types of stressors. And furthermore, just to give some credence to some of the evolutionary work that I've done, by modeling kind of past evolutionary history and diversification, we might be able to project that into the future and understand how traits that species have or already have might be able to evolve or not to future climate change scenarios. So in conclusion, genomics has transformed our understanding of global biodiversity and helps us to conserve endangered species, helps us to even understand what species are out there and understand their relationships. But I think that there are also techniques that we can use to apply these to ask questions really about solving some of these bigger challenges, such as climate change and epidemics, and how biodiversity is a fundamental part of that whole planetary kind of health. So while I haven't much worked in that kind of like public health and climate change space yet, I'm really excited for opportunities to potentially kind of expand biodiversity genomics into these spaces. So thank you very much.
Thank you, Dr. Tan. Oh my gosh, this notion of how the origins of biodiversity and this and genetic expression and just how these external factors may impact it. It's just fascinating, and your work seems just like cutting edge. And how exciting that perhaps you can use some modeling for predictions that would impact, planetary health. So thank you so much. What an interesting perspective. And we really appreciate your comments and just the ideas, thoughts for collaboration. And it's very nice to meet you. So thank you so much. I am going to move on to Professor Paul Brown. So Dr. Brown is a professor of molecular biology and the head of the Department of Basic Medical Sciences at the University of the West Indies. And he holds a master's in philosophy and biochemistry and a PhD in microbiology from the University of the West Indies. He's involved in several research projects spanning public health-related infectious disease and basic sciences. He's currently on the Council of Microbial Sciences and the American Society for Microbiology, and he previously served as vice chair in 2017. He's also active with has been a council member at the International Society for Infectious Diseases. So we're very excited to have Dr. Brown. Thank you.
All right, thank you very much. Thank you very much, Dr. Holly. Just check everything is okay. Great. You're seeing my slide. All right, so thanks to the organizers for having me as part of this important roundtable discussion series. My contribution to the EPI portion is rooted in the concept of follow that microbe to highlight work that I have done and others that are in train. As a microbial scientist, and for the purposes of this talk, for better or worse, I feel that epidemiology is really about the microbe and the conditions that facilitate its ability to cause disease in susceptible populations. So in the case of Leptospira, the microbe that causes leptospirosis, its epidemiology reflects the ecologic relationships with humans and chronically infected reservoirs, whether these are domestic, stray, or feral. These relationships can be occupational, recreational, or accidental, and are usually seasonal in occurrence, but more associated with the second rainy season or rainy period in Jamaica. We have a kappa of 0.9 for this association in the second rainy period in Jamaica. Studies with the Climate Research Group have shown that climate factors can explain close to 75% of cases, and El Nino is closely tied to cases. Interestingly though, La Nina, where you have overall increased rainfall, causes more cases throughout the year. Since symptoms of leptospiral infections can mirror that of dengue and malaria, and we heard a lot about malaria earlier, and other diseases, cases can be missed, for example, during a dengue outbreak when there were cases of both, but only one is sent for a confirmatory diagnosis, or mixed, as in this study, which found co-infection in about 1 to 2% of cases. But just to note that exposure to dengue and malaria appear to occur more frequently among children, and Prof. Celia would have noted that in relation to dengue, while leptospirosis was more common among adults. For better or worse, the organism can attach to various cells and cellular structures, which allow for colonization of tissues during the infection process. Interestingly, there is a decreased attachment in the presence of fucose and mannose, something that is similar seen in E. coli and urinary tract infection using mannose as a sugar to block that interaction. And one strain of the microbe, or Petersonii, Serovar Jules, appeared to be more virulent than another, Interrogant Serovar Portland Ver. Together, these two endemic serovars account for most of the cases of leptospirosis in Jamaica, with Jules tending to be spread between animal and human, that is a more direct transfer, while Portland Ver is more present in a contaminated environment. And the rats get a bad rap for this, but just to say that rats are only one of the important causes of leptospirosis in Jamaica. Definitely for worse, temperature and oxidative stress appear to be triggers for virulence in these microbes. Again, with Portland Ver being more responsive to the stresses. And I think there was some discussion about the effect of climate change and what is happening with infections. The genes studied included those associated with attaching to surfaces, evading host defenses, invading host tissues, cell hemolysis, and iron homeostasis. While leptospira have two genomes, yes, two genomes, the differences observed are likely due to the genetic differences. Portland Ver has a larger genome found in the environment. And with climate change, generally higher temperatures, both of these serovars are primed for increased virulence well before they contact a susceptible host. So the microbe can cause a myriad of symptoms. In a Bayesian study that formed a part of my PhD thesis, the most notable symptoms were jaundice, headache, dysuria, acute renal failure, now called acute kidney disease, proteinuria and thrombocytopenia, low platelet count. Serological analysis was not helpful for these patients. They were all diagnosed at postmortem. And we carried out similar studies in Jamaica and found fever, muscle and joint pain, nausea, vomiting, abdominal pain, blood in the urine, and thrombocytopenia as the main symptoms. In households where there were cases, a small but significant number of water samples were positive for leptospiral DNA. In that study, residents in rural communities, the presence of garbage dump and leptospiral DNA in water samples correlated with households with a history of the disease. And this was actually very statistically significant at the 0.01 level. The microbe is known to thrive in tropical and subtropical areas that are classified as slums, inner city or rural areas. Michael Marmot, chair of the Commission on Social Determinants of Health in WHO, said some time ago, what good does it do to treat people's illnesses and then send them back to the conditions that made them sick? In fact, the fact is that 1 billion individuals, representing about 32% of the world's urban population, live in slums. Further, the UN Millennium Declaration pledged to achieve significant improvement in the lives of at least 100 million slum dwellers by the year 2020. Clearly, this was not achieved. Unfortunately, the world's urban slum population will double to 2 billion in the next 25 years. Going back to the Barbadian study, multi-system involvement was indicated based on successful detection of the microbe in more than one tissue sample, which corroborated with the clinical and pathological findings of the 8 fatal cases that were studied. We found the microbe mainly in the CSF and brain, among other organs and tissues. And we use immunofluorescence and PCR as well as culture for detection. In the Jamaican study, the main organs or tissues where the microbe was found at postmortem were in the heart, lungs, and eyes. And these results suggest that in acute and/or fatal leptospirosis, the pathogenesis and pathologic features are related to the presence of the organisms in the tissues. And for those who are wondering, the leptospira is a spirochete, and it follows a similar trend to other spirochetes like Borrelia and Treponema, which actually has some incursion into the central nervous system and the brain sometime in the disease. The interesting thing with these cases is that all of these incursion into the brain actually occurred within 1 week post-infection. And in the case of the Barbadian study, all of those patients died within a week. Within 6 days, within a week of admission to hospital. And again, because these cases are usually diagnosed late. So in closing, and Dr. Holly, I am very particular about the time, so I didn't want to go over time. So we have been doing work looking at gut microbiome in health and disease, and we looked at 42 healthy undergrad Faculty of Science and Technology students. We didn't find any major differences in that study, and this was quite shocking to the Trinidadian students who realized that they were not different, at least their gut microbiome, from their Jamaican counterparts. We recently got funded a D43 grant to look at periodontitis and associated systemic comorbidities in children and adults, and that work is starting. We have been looking at mixed species biofilm cultures and asking the question: is the interaction antagonistic, mutualistic, or synergistic? And we have been looking also at heavy metal and disinfectant resistance. We are finding that 2 or 3 organisms can actually cooperate even though they don't like each other. We want to look at STEM, that's transmission, and EM, that's electron micrograph for biofilm analysis in mixed cultures. And also, I think Dr. Wilson mentioned something about this antimicrobial resistance, bacteria and genes in sewage, wastewater, water catchment facilities. As this is a new era in Jamaica, I think it is quite important that we actually get a handle on what is happening in our wastewater systems. So with that, I close, and thank you very much. And for further questions, comments, you can email me, or I can be found on X as well. Thank you very much.
Dr. Brown, thank you so much for your comments. I think what was especially compelling, your comment about you treat a disease and then send people back into the same risk factors, the social determinants, and then just this notion of the multi-system nature of leptospirosis. And it was something that we try to teach our students, is to recognize emerging diseases that we're not used to seeing and recognizing atypical presentations. And some of your future work and the work with wastewater is really compelling, and there's just such a huge role for it. So thank you so much. It's scary, but the optimism is that someone like you is working on it. So thank you so much. We're just coming to our last speaker. We're so excited to have, our speaker hero. I'm going back to Dr. Royce. Our last hero of the day is Dr. Brian Allen. Dr. Allen is the Associate Director for Academic Affairs at the School of Integrated Biology, and he's a professor of entomology at the University of Illinois. And his work includes, there's so much, I'm seeing so much overlap now. His work includes ecology of infectious diseases, especially those transmitted via infected arthropods. And much of his research focuses on the consequences of human-mediated global change, such as climate change and human land use, and on the risk of exposure to parasites and pathogens that are carried by wildlife. His interest in impacts of landscape change and emergence of tick-borne diseases is part of that. And he uses numerous tools, including molecular technologies, remote sensing, and theoretical modeling. So, clearly we see, as I come to the last hero speaker, we see this sort of biodiversity, the infectious disease components, the land use, the nature-based solutions. There's so many, every topic that has been discussed, and I just see such potential for collaboration. Anyway, Dr. Allen, please take it away.
Thank you, Dr. Rosenkrantz. I really appreciate that introduction. And thank you, Dr. Brown. I really enjoyed your talk, a lot of similar interests. I work on a variety of different topics in the biology of vector-borne diseases, but I thought one possible point of common interest could be in mosquito control, and particularly how stormwater infrastructure and stormwater management impacts mosquito control. So I'm just going to give a brief overview of some of the research I've done on this topic, and here's a list of collaborators in this research. I couldn't do this without their help. Here in the central Midwest, this is our typical stormwater management system. So along roadsides, we have a subterranean stormwater catchment system. It's designed to capture precipitation during a rainwater event. And for 99.9% of people, that's the extent to which they think about stormwater management. Stormwater engineers and mosquito biologists are two groups of people who think about this a lot more, because there's a lot of action happening beneath the surface, and that's where things get interesting. So on the right here, you can see a diagram of a typical catch basin sump. So this is what is below that catch basin grate along the side of the road. And these are an old design for capturing stormwater runoff during a stormwater event, and there's often a connection to some kind of sewer system, which also raises a number of issues. But one of the things that is problematic with these catch basin sumps, most Midwestern and Eastern cities in the US have this type of design, is that they become habitats for mosquitoes. So especially mosquitoes in the genus Culex, females will fly in through the catch basin grate, lay their eggs, and these are actually ideal habitats for their larvae to develop because there's a large amount of debris in the basin. So there's a lot of bacterial growth, microbial growth for those larvae to feed on while they develop. A very interesting phenomenon in North America is that we tend to get outbreaks of mosquito-borne disease during periods of drought, which is the opposite of what is intuitive for many people when they think about mosquitoes and their larval habitats. But the reason it's strongly associated with drought is because these catch basin habitats actually become more stable during drought events. That connection to the sewer is this actually kind of key idea in this concept, that when you get a lot of rain, these catch basins flush and they flush the larvae out into the sewer system. But during periods of drought, the water level drops below that connection to the sewer, and these have become more stable habitats. So in the eastern US, we get big outbreaks of West Nile virus, which are transmitted by Culex species mosquitoes during periods of drought because of this catch basin design. For reasons unrelated to mosquito biology, stormwater engineers have been trying to come up with more environmentally friendly ways to manage stormwater to improve water quality. And so we're experiencing this explosion in technologies that are called green stormwater infrastructure. They include things like green roofs, permeable pavement and parking lots, rain gardens, and something that's widely used in other parts of the world, which are rain barrels, which are a domestic form of green stormwater infrastructure where people can capture excess rainwater runoff and use it for watering their gardens. And so one of the interesting questions in mosquito biology is what's going to happen as municipalities increasingly implement these other types of technologies, often where stormwater is infiltrated on site. And so instead of capturing and holding rainwater, these green designs are meant to mimic natural hydrology and allow that stormwater to infiltrate down into the soil. And as a result, water may be impounded for less time. In my lab, we've conducted 3 studies to look at different types of green stormwater infrastructure and understand its impact on mosquito production. This is work led by my former postdoc, Andrew Mackay, who's now an associate research scientist at the Illinois Natural History Survey. And I think he presented in the last discussion held by this group. One of the first green stormwater designs we tested was comparing conventional catch basins, like the one I described previously, to a new technology called infiltration catch basins. And infiltration catch basins are a very similar design. They just have a permeable bottom. So instead of capturing and holding rainwater, they allow it to infiltrate into the soil. We took advantage of a natural experiment, which is that a city near here, the city of Aurora, was replacing conventional catch basins with infiltration catch basins. And so we took advantage of that municipal project and identified a control area nearby where they were not replacing catch basins and compared what happened before and after the implementation of green stormwater infrastructure. What you can see on the top panel there is the proportion of catch basins holding water. And in green are the catch basins that eventually became infiltration catch basins starting in 2014. As you can see in 2013, those conventional catch basins held just as much water as any other catch basin. But when they were replaced with the infiltration catch basin, they were much less likely to hold water than conventional designs. On the bottom panel is the number of mosquito larvae that we sampled in those catch basins. And as you can see, the catch basins that in 2013 that eventually became infiltration catch basins actually were some of the most productive catch basins for mosquitoes. And once they were replaced with infiltration catch basins, we almost never detected mosquito larvae. Those catch basins were almost always dry. And so here seems to be one example where the implementation of a green stormwater tool reduced mosquito production in an urban environment. A second study we conducted looked also in the city of Aurora, where they were implementing, in addition to these infiltration catch basins, rain gardens as another form of green infrastructure tool. For the sake of time, I won't go into all the details about what was happening here, but these rain gardens also had catch basins associated with them. And we saw a very different but interesting outcome in the case of rain gardens, where those catch basins associated with the rain gardens were still very likely to hold water. So there was a lot of water going into the rain garden that was finding its way into these catch basins, but the water had flowed through the soil and the plants in the rain garden before getting into the catch basin. And so the water tended to be cleaner. There were fewer nutrients in the catch basins associated with a rain garden. And the measurements we took on mosquitoes that were emerging from those catch basins showed that they tended to be smaller and with less survival potential. And so as a result of having the water first flow through a rain garden that removes a lot of the nutrients from the water before it ends up in the catch basin, mosquitoes were still colonizing that habitat, but those tended to be less fit mosquitoes. So a second example of where a green infrastructure tool could actually reduce mosquito production. Finally, a third type of green infrastructure we looked at is the residential tool of rain barrels. And here appears to be an example of where a green infrastructure tool can actually increase mosquito production. We sampled rain barrels associated with people's homes across an entire summer, starting in June and into September. And we found over 50% of the rain barrels had mosquito larvae present on at least one occasion. As the summer went on, the community of mosquitoes became dominated by one of the invasive species that's common in this region and throughout the world, the Asian tiger mosquito, Aedes albopictus. And what this suggested is that poor management of this particular type of green infrastructure tool increased mosquito production. That's just a summary then of our findings. And so I can wrap up there for the sake of time. And so Dr. Rosencrans, I'll hand it back to you.
My gosh. Oh, thank you so much. That, just fascinating. This notion of infrastructure, rainwater infrastructure, is new in my vocabulary, and the notion of these infiltration catch basins. I do see, and that's, we talk about climate change, and standing water is we talk about impacts from water. We think often about infectious diseases, but the standing water and ways to mitigate is just such a novel approach. And thank you so much. Fantastic. I'm so grateful. Well, I'm going to, just for sake of time, I want to close up the session, but, we've clearly delved into intricate relationships between vectors, habitats, human well-being, infectious diseases. And, I just texted my colleague, what an impressive group of people. We've featured such accomplished speakers from University of Illinois and University of the West Indies. And so it's evident that there's a true intersection of entomology, environment, and health. And I think this just presents such a vast landscape of knowledge and the potential for impactful research and collaboration. So it's been just I think we're kind of want to leave everybody with an optimistic message as we step away from this forum. Love this idea of the spirit of collaboration and inquiry, and taking our collective expertise to foster positive changes in our communities. And I think we're all trying to forge a journey toward a healthier, more sustainable future. And that's how we're going to do it, through this continued dialogue. To chart a way forward. So I just want to express my deepest gratitude to our distinguished speakers. I'm going to steal Dr. Roy. I'm stealing heroes from you. I'm also coming to Jamaica but for sharing expertise and to just everyone for your engagement, for the preparation you did to create these wonderful presentations. And I hope that these discussions are catalysts for future endeavors. Thank you all. And thank you again for the privilege of being a moderator. And I have, and that is the end of my comments. Thank you.
Some persons have been reaching out.
I know that for myself, health connection is a big part of what we're doing. And it's not just the food, but it's just microbiomes on a whole. So that really intersects with some of what Dr. Paul Brown talked about. Dr. Christie, some of your work on these vector-borne diseases certainly is one of the human health impacts of climate change that Dr. Rosencrantz and I and other people are very much interested in. So there's just, to me already, just from this session, so much potential for collaboration. So stay tuned, everybody. And I think I can sort of speak on a little bit on behalf of the university, because our vice provost, who opened up the session, was here because they're very excited about the idea of UIUC forging collaborations with the University of the West Indies. And as a graduate of the University of the West Indies, I could not be prouder or more thrilled about that potential moving forward. And I know that UIUC is very much interested in forging collaborations and partnerships with the global South. You know, it's one thing to have collaborations with, you know, the kind of first world developed universities with but it's but they really want to have an emphasis on the global South, and University of the West Indies definitely fits that. And we're kind of in each other's backyards. So I hope that we will see more to come. Dr. Busby Earle has just been a powerhouse in organizing. So I just want to give him a big shout out for that.
You have my support and the support of the pediatricians and the virologists. You heard from Dr. Anzinger, who is the head of the microbiology department last time.
Yes.
And I'm sure we have, you know, his support because we all work together.
Exactly.
And you also heard some additional comments on the other viruses, leptospirosis and so on, as Professor Brown just said.
Yes, absolutely. And they're like my husband, Dave Ram Kumar, who's also a graduate of UE Med School, wasn't able to participate, but he's very much involved in all of the work that we do. So stay tuned. He will definitely be involved in future endeavors.
Okay. Thank you all very much. Thank you very much for those kind words, Jafia. Dr. Rosenkrantz, champion. Very, very, very nicely kept everyone on their toes in the time. I really appreciate it. And of course,
I felt so bad cutting anybody off, and I often was
I
doing it
know.
on their last slide, and I'm like, oh, if I just, but I hope I think